Hey, what's going on, people? My name is Matthew Jose, and today I'm bringing you the physiology of the endocrine system. I haven't uploaded a video in a very long time, and that's mainly due to me being ill and I haven't been able to do work lately. I've done the anatomy videos, it's just I haven't uploaded them as of yet. But in this series, I will be doing the physiology of the endocrine, as you can see here on the right. My name is Matthew Jose. You can search me up on Facebook and whatnot. So let's get started. So this video here is going to be the basic endocrine system and what the endocrine system is. So it's not going to be the different types of hormones or whatnot. So it's just going to lead you up and give you a basic idea of what endocrinology, what the study of it is, and then from next video onwards we can start to look at different hormones and how it acts on target cells and so on and so on. Right. So we know basic endocrinology. Endocrine system is basically different methods of communication within the endocrine system, and these include, as you can see here on the PowerPoint slide, autocrine mechanisms. Autocrine is basically the simplest and the shortest way of communication within the endocrine system, and it's basically it operates over a very short distance. And here you have a host cell, and what happens is it releases a messenger molecule, which is shown by the arrow here, or you can follow my mouse. So a messenger molecule which acts on the host cell itself. So it's the cell which releases messengers which acts on itself, and that's autocrine. So it's auto-regulating itself. Paracrine is another method in which these cells release messengers which acts on neighboring cells or cells within the close vicinity. So you can see here, this cell here releases the messenger molecule, and this then goes on and acts on neighboring cells. It doesn't have to be next door, but it can be, in a close vicinity, within a close proximity, or a parentheses, you can say. Now, the endocrine system is what we're most interested in, and the way in which most of our body works, and it's the longest distance communication method, and it usually involves a cell which releases a messenger molecule that then travels into the blood circulation, so via your blood vessel, and then it travels through the blood vessel and then it reaches a target cell. Right, and that's what we're going to be focusing on in the next few slides and so on. So here's a method. So here's another simpler view. So endocrine cell, this is the starting cell here, which I'm pointing to now. That cell releases a messenger molecule. This then travels through the blood circulation, and when it travels through the blood circulation, you can have various different things, such as it can bind proteins, and then it undergoes metabolism when it reaches the liver, and then from the blood circulation it then reaches target cells, where it acts on target cells via specific receptors. These receptors can either be membrane receptors or cytosolic receptors, which I'll go into a bit more detail. And examples of target cells include things like skeletal muscle cells, brain cells, liver cells, pancreatic cells, and it's mainly the liver cells that's mainly the thing, but the skeletal muscle cells are quite a big form a big part of the target cell as well. Alright, so the basic model of this is you have an endocrine. It releases a messenger molecule which travels through. There's this red thing here, the messenger, through the blood vessel, through the circulation, and then acts on a target cell to bring about a physiological or a biological response. And I'll give you an example of that. Now, there are problems associated with the endocrine system. For example, when the messenger molecule is secreted into the blood circulation, what happens is two things that can happen, mainly happen. One, the messenger molecule could become diluted within the blood plasma. Two, it can bind to plasma proteins like albumin, glycoprotein, and so on. Three, it can be inactivated when passing through the liver due to metabolism. Right, now all of these three things mean that the messenger molecule there wouldn't be adequate concentration of the messenger molecule reaching the target cell. So therefore, this would mean that it wouldn't be able to produce a desired effect. The only way to do this would be to increase messenger secretion, right? So it means that if the endocrine cells constantly summate together, so if you know the nervous system, or you've done A-level, nervous system, you know what spatial and summation is. That's when one cell constantly releases messenger molecules, or releases molecules in subsequent or adequate manner. Now, what happens is the endocrine cells cannot do that. Instead, what happens is endocrine cells, loads of them come together or clump together to form what's known as glands. And we have various different glands around the body. We have our sweat gland, which is located under your armpits. That's an example of gland. We have various different types of other glands within the body, and this is basically a gland, and is basically loads of endocrine cells that's packed or clumped together. Now, when there's loads of them and there's a gland, when all of these secrete endocrine at one go, it means it's secreting a very high concentration because there's a large amount, and therefore when passing through the blood vessel, there will be a very little amount that gets diluted, which binds plasma proteins and then becomes inactivated due to metabolism. But there'll be an adequate concentration of them which reaches the target cell. Now, so how does it all work? Right, so all of a sudden your endocrine cell, or your initiator progenitor cell in this case, doesn't just have this spark of an idea where it wants to release hormones, hormones is another name for soluble messengers. What happens is you usually have a stimulus. The stimulus then causes the endocrine cell to release the soluble messenger, which in this case could be hormones, which then travel through blood circulation and then acts on target cell. And we also have a negative or a positive feedback related to this. So an example is given on the right here. Stimulus could be, for example, if you have a heavy meal large in glucose or sugary content, what would happen is your plasma glucose concentration would shoot right above 7 millimolar. The average normal range would be about 5 to 6, and this will shoot up to around 7, 8. This rise in plasma glucose concentration is usually detected by these cells here, this initial endocrine cell, and these can include things like beta cells found within the pancreas or the islet of Langerhans, or alpha cells which releases glucagon. So an example of an endocrine cell here would be beta cell, right? Now the stimulus would be increase in plasma concentration. Now the beta cell detects an increase in plasma concentration, therefore it releases a soluble messenger, which is known as a hormone. In this case, we all know this very well, and this hormone is known as insulin. Once insulin is blood-bound, so it gets into the blood circulation, it travels through the blood circulation and it reaches target cells. Insulin then reaches target cells like skeletal cells, liver cells, brain cells, which acts to remove glucose. So they bring about a biological response, and this could be to take up more glucose, or the liver could convert some glucose into glycogen and store it for later use, and various different things. It can increase the body's metabolic function, so therefore you're metabolizing more, so therefore you require glucose for respiration, so it takes up the glucose from the plasma concentration. Now, as this process goes along, the plasma concentration of the glucose eventually drops. So therefore, once it gets to a norm, what happens is a negative feedback. So the negative feedback means that the endocrine cell is no longer stimulating any more release of insulin. Stimulus is defined as anything that can cause a change in the normal levels of the environment. Now, there are different families of hormones that we need to know about. and I've listed a few of them, the main three ones here, and what their properties are here on the right. So the first one that you want to know is peptides and proteins. Examples of these include insulin, angiotensins, and these are basically, if you look over here, they're hydrophilic, so they're water-loving. So thus, they do not diffuse across the cell membrane because they don't want to, leave the water behind, so they are hydrophilic. The next set of examples are amino acid derivatives. These include thyroxine and adrenaline. Adrenaline behaves more like the peptide and proteins, so therefore it has to be more hydrophilic, whereas thyroxine is more hydrophobic. Then we have steroids, and examples of these are estrogen, corticosteroids, or cortisol. They're hydrophobic, which means they're water hating. Now, thus they freely diffuse across cell membranes. So these are the main three types, and examples of these are given here as well. So peptides and proteins examples: insulin, steroids is estrogen, cortisol, amino acid derivatives is adrenaline or thyroxine. Now, a bit more information for the keynotes. I'm a keynote myself, I think. So, in terms of peptides, proteins, and hormones, they contain a lot of carboxyl and amino acid groups. Many will be ionized at physiological pH, and therefore these hormones are typically very hydrophilic. So the main point that you want to take from here is, due to the fact that they have a very large number of carboxyl and amino acid groups, therefore that means that they can be ionized at physiological pH, and therefore these means they're hydrophilic, so they're water-loving. Now the amino acid derivatives like thyroxine, like I said before, behaves more like a steroid, whereas the adrenaline behaves more like a peptide hormone. In the case of steroids, they have a cholesterol backbone. That's the main thing that you want to understand. They have a cholesterol backbone and they're very hydrophobic. So this is basically an outline of what happens when peptides and proteins. Examples of peptides and proteins. What happens when they're released. So initially, what you have is your progenitor, or the first cell that secretes these molecules. These molecules then travel through your blood circulation and into your target cell. So how does this do these? So what you see on the screen now is very simple procedure. This is not the the in-depth level. The in-depth level was, I'll go into a bit more detail as we go along through the course of the endocrine physiology. So first, what happens is the enzyme, the hormone or the soluble messenger molecule is encoded by genes within the nucleus, and that's then stored. It goes through the Golgi apparatus and the smooth endoplasmic reticulum, and then it's stored in vesicles, and then upon calcium activation, or this is Ca2 plus independent release, these molecules are released. They then travel through the circulation, and bearing in mind that peptides and proteins are hydrophilic, so they have water loving, so therefore they don't easily diffuse through the membrane, the plasma membrane, so therefore they have membrane receptors on the surface. And examples of these membrane receptors you include receptor tyrosine kinases and G protein couple receptors. And then when they bind to these membrane receptors, they then cause secondary messenger molecules like cyclic AMP, which activates various other enzymes on the way, or a cascade, you can say a cascade of reactions, which then bring about a biological response. So peptides and proteins, they're hydrophilic messengers. Growth factors are an example. So here we have G protein couple receptors, and here we have receptor tyrosine kinases, and insulin is something that works via this, or growth factor receptors. Now, before we go into how does GPCR work, we're going to go back. So, sorry, computer's a bit slow. Right, so, no, actually we're going to go forward and then back to make it a bit more clear. How does a GPCR look, and how does it work? So, the G protein coupled receptor looks something like this. So it has a receptor binding site, which is shown here, which has an extracellular domain, an intracellular domain, or you can say cytosolic domain, and a transmembrane domain. Okay, so it has all three, and it's almost like this cylindrical shape. Or before, back in the olden days, they used to draw it like a squiggly line going up through, you know, and then towards close in proximal in very close proximity, they have what these three subunits, which are also part of these G protein coupled receptors, and these are the first messenger molecules in a sense, you can say, and they contain guanine alpha subunit, which is bound to a GDP, so diphosphate, guanine diphosphate, and a beta and a gamma. So what happens is what you want to take note of is this cycle here. So moving along with the mouse here. So upon stimulation, the basic thing that you want to know is initially when a stimulus is bound to the G protein coupled receptor, the G alpha subunit, so the big circle, the big blue circle, detaches from the beta and the gamma subunit, and the GDP on it is replaced by GTP, causing it to be turned on. Right, so you can see here, this diagram is slightly complicated, so you might want to listen up very carefully on how to understand it, and to make sure you understand it very carefully. So what you have here is GDP in the red, and you have GTP. So you see GTP is the on format. So it's only when GTP is bound to the guanine alpha subunit that it becomes in an on state or in a working condition. Whereas normally when the G alpha subunit is bound to the beta and gamma, it has a GDP bound to it, and it's usually in the off state. So how does it become on? Well, it does this by a molecule called GEF, guanine guanine E factor. Okay, so this molecule here corresponds to this bit here. Now, when GEF is activated, it activates a protein kinase, and protein kinase's job is to add a phosphate molecule to any group. Right, so you can see here is diphosphate, and this is triphosphate, so that clearly this is an addition of a phosphate. And the ATP, the phosphate comes from an ATP molecule. So the GEF, what you need to remember is the GEF part, which activates protein kinase, which then causes GTP to be turned on. Right, now one other thing is, okay, once its function has once it's turned on and you want to turn it off, the enzyme phosphatase is what removes the phosphate group. So that this part here corresponds to this area here, and that phosphatase enzyme is activated by a molecule known as GAP, guanine activating phosphatase. Okay, so GAP, all you need to know is GEF and GAP. So GAP turns off the GTP, it turns GTP into GDP, whereas GEF turns GDP to GTP. Right, so hopefully you understand it. So this diagram here is slightly in the opposite direction, because this side here, where my mouse is pointing now, corresponds to this area here, whereas this bottom bit here corresponds to this top bit here. So don't, if you didn't quite understand that, you might want to rewind back and listen to it again. Make sure you understand it. So once it's in the activate state, what happens now is it activates a secondary messenger molecule like adenyl cyclase, which then goes on to activate cyclic AMP, which you all would have learned about in A levels and stuff, which then goes on and activates pKa, and then so on and so on until it reaches the nucleus and causes a gene expression or brings about a biological response. Now, in terms of insulin, insulin works through this process. So initially what happens is insulin, so in this case insulin is a ligand here. It binds onto receptor tyrosine kinase, and it forms a dimer. So the phosphatase comes from the ATP molecule here. Now, once that becomes activated, this then causes a molecule called GRB to be activated, which then causes SOS to be activated, which then activates a RAS system, which is here. The RAS then goes on to activate REF then goes on to activate MEK pathway, and this whole pathway is known as the MAP kinase pathway. Right, this then travels in, so all of this has taken place. Okay, this happens within the membrane receptor, because remember these are hydrophilic messengers. Right, now all of these mechanisms here, so the GRB, SOS, RAS, REF, MEK, and then down to MAP kinase, all of that happens within the cytosol. Now, once MAP kinase is formed, they then travel across the nucleus, so it crosses the nuclear membrane and then into the nucleus, where it causes transcriptional response to bring about biological responses. Now, steroids was in the other family of hormones that we looked at. What happens here is enzyme activity causes steroid hormones to be released. These then travel through the blood and then binds to proteins. They then go on to target cells. Now, the target cells works via, because these ones are hydrophobic, they are easily diffusible through the membrane. Right, so most of their receptors are cytosolic or nuclear receptors, so receptors that are found within the cytoplasm of the cell, or receptors that are found within the nucleus. An example of this will be steroids, cortisol, so cytosolic nuclear receptor examples will be estradiol, cortisol, adrenogen receptors, and so on. And so hopefully from this video here you've got a basic idea of what the endocrine system is and how all of these works. Now, in the next video I'm going to go through a bit more detail on the different classes and different sections and different parts of the endocrine system and how each one works. So anyway, so if you guys enjoyed the video so far, please do leave a comment, like, subscribe. It really helps the channel. Once again, it's been a pleasure teaching you. I hope you benefit from it, and please do subscribe to the channel. My name is Muffin Jose, and thank you for watching.